Can you open up to page 173? Open up to page 173. And you have a model, a flat model with a longitudinal section of the kidney, and in the middle you have the one single cortex and medulla showing you a cortical nephron and a juxtamedullary nephron. And the last model on the far, what that would be, the tiny one, that is the renal corpuscle with the glomerulus showing you the parietal layer and the visceral layer, the podocytes. Okay? Now, we have gone through the anatomy in pretty good detail. So now, your job is to start identifying. So, if you open up to 173, I will tell you the structures that you are going to identify. Kidney anatomy. Okay? Renal fascia, you cannot see it on the model because that's an external connective tissue. You can't see the fat, which is the adipose capsule, but the outer wall of the kidney is the renal capsule. So check the renal capsule. You can identify that. Hilum, you know, which is the hilum, the part where the ureter and the blood vessels enter. Okay. Then number 3, try and identify all of them. Renal cortex, renal medulla, renal column, renal pyramid, renal papilla, minor calyx, major calyx, renal pelvis. Okay, so try to label all of them. I'll try and get you some more tape for labeling. And then the middle panel is your structure of the nephron. So go down to the structure of the nephron. Identify renal corpuscle, glomerulus, glomerular Bowman's capsule, afferent arteriole, efferent arteriole, peritubular capillaries, vasa recta, there's no need of that, Bowman's capsule, glomerular capsule or Bowman's capsule, parietal layer, visceral layer, podocytes, capsule space, all 4. And then number 2, renal tubule, proximal tubule, nephron loop, descending limb, ascending limb, distal tubule. Okay. Juxtaglomerular apparatus, JG cells, that is on the afferent arteriole, you can see it, macula densa cells, and then collecting duct. Collecting duct. You can't see the papillary duct. Papillary duct are just, collecting ducts will drain into papillary ducts, but collecting duct, okay? So that is what you would have to identify, okay? Now, on this model that it's not there on the first table, but the model that you have, not this one, that one, yes. So start with this model, please. Start with that flat model, okay, the big one. On this model, which is there, one in each table, this model, you might have to use this. So on this model, identify the bottom portion, ureter, urinary bladder, urinary bladder, and then detrusor muscle. Detrusor muscle is a muscle, smooth muscle of the urinary bladder is called detrusor muscle. Urethral orifices, opening of the ureter inside the bladder. Trigone, internal urethral orifice. So, other urinary structures, identify 1 and 2, A, B, C, D. And urethra you can identify. I don't think the model shows the internal urethral sphincter, external urethral sphincter, just the urethra. Okay? So kidney anatomy, structure of the nephron, other urinary structures, except the blood supply. The blood supply, we will go over it. I will show it to you and then you can identify it on the model. So go ahead, whatever you learned this morning, if you have your notes, well and good. Or I also have the urinary system lab on Canvas. You can also open that. You can use your pictures that are there in your lab manual to also identify. Okay, so please start. So that there are two, we are going to look at the blood supply, and then there is also one more, we are going to look at some microscopic structures, and there's one last thing which is urinary system physiology. We are going to analyze some fake urine samples, okay, using reagent strips. So that's the last part that we have to do. So once you finish the anatomy, we can go ahead with that. I will get you some tape, and then you can, whatever works for you. So we have some tapes here, and if that's something that you want to do. So please pay attention, look at all the parts, because next Monday is your lab exam on respiratory system and urinary system. So make sure that you look at these. Plus proximal convoluted tubule, nephron loop, nephron loop, okay, that is the descending limb and the ascending limb. So you should, if you don't know which one, you have to trace it. If you trace it, you will be able to see which one is going down, which one is going back up. And then distal convoluted tubule, collecting duct, and then the opening of the collecting duct is the renal papilla. Now, on the same model, you should know afferent arteriole. So look on the right side of the picture, afferent arteriole, efferent arteriole, renal corpuscle, arcuate artery, arcuate vein. We are going to come to this. Okay, so the middle one, the nephron structure is important. You should have identified all the parts of the nephron: glomerulus, Bowman's capsule, PCT, descending limb, ascending limb, DCT, collecting duct. You should have identified all of them. Afferent arteriole, efferent arteriole. Okay. Now try to look at the blood vessels. For the blood vessels, you can use that first model. Okay, you can use the first model. Now here, this is primarily memorizing the terms. Okay, so you are able to see the blood vessels on page 166 of your lab manual. So let me tell you how blood flows into the kidney. So the kidney, both kidneys receive about 20 to 25% of the cardiac output. Very important organ, it is filtering the blood. So, the cardiac output is about 5 liters per minute. So almost 1.5 to 2 liters per minute goes to the kidney. Okay. So renal artery, which is a branch of the aorta. Remember we studied the branches of the aorta, right and left renal artery. So renal artery enters, this region is called the hilum. So this is where it enters. As soon as it enters, it splits. Okay, it splits into 2 or 3 branches. So the first one is called the segmental artery. Segmental, because it's going to each segment of the kidney. So you may have about 2 or 3 of them. Then the segmental artery then travels in between the pyramids. So you're going to see the artery in between the pyramids. The name changes. Okay, so these are again branches of the segmental artery. So the segmental artery enters the kidney and then it splits again. Each branch will go in between the pyramids. Now this is called the interlobar artery, interlobar. Because if you consider this each a lobe of the kidney, so in between the pyramids, interlobar artery. Then the blood flows, this branch will then go and curve on top of the pyramid like an arch. Like an arch. It's called the arcuate artery. Arcuate artery. So this is the way blood flows. Renal artery, segmental artery, interlobar artery, arcuate artery, and from the arcuate artery you're going to see all these long branches going into the cortex. Those branches that you see. These are called cortical radiate artery because they are in the cortex and they look like the rays of the sun radiating. So it's called cortical radiate artery. The old name was interlobular artery. Interlobar, interlobular. You can remember either one of them. In the exam, I'll have both those names. Okay, so you can remember either or. Whichever is easier for you, but in the exam, I will give both the names so that you're not confused. Okay, so interlobular artery or the cortical radiate artery, it is from this that the afferent arteriole branches. So the afferent arteriole that you're seeing here in the middle model, so this afferent arteriole that you're seeing is coming from this cortical radiate artery. It's a branch of that. Then you have the glomerulus, efferent arteriole. The efferent arteriole, once it comes out of the glomerulus, it further splits into capillaries which surround the renal tubule. These are called peritubular capillaries. So that's why in the lecture I told you the kidney is slightly different because it has two capillary beds, two capillaries. One is glomerulus and again peritubular capillary, and after that it is the venous side. Okay, the venules. The veins have the same name as the artery, same names. Okay, so you're going to see peritubular capillaries, interlobar vein, arcuate vein, this is the cortical radiate veins, arcuate veins, interlobar vein, segmental vein, renal vein. Renal vein drains into the inferior vena cava and goes back. So the pathway of blood flow is like this. This is the pathway of blood flow. Okay. This is the oxygenated side right up till this peritubular capillaries, and after that it's all veins. Okay. So try to identify them on the model as well. Okay. So you should be able to identify all of the ones that are listed. So if you look at page 1, the list where we had 173, number 4, blood supply, identify renal artery, segmental artery, interlobar artery, arcuate, cortical radiate artery, afferent and efferent arteriole, peritubular capillaries. Okay, don't worry about vasa recta. Renal vein, you should be able to identify renal vein, interlobular vein or cortical radiate vein, arcuate vein, interlobar vein. Okay, so that's the blood supply. So if you have not done that, do the blood supply as well. do the blood supply as well. Make sure that of course you have to go back and read this. It's not going to come in this class. But at least make sure you're able to identify. And then we are going to do the renal physiology. We're going to do the urine analysis. Open up to page 175. I have actually focused. Look at 175, figure B. Figure B. Okay? So this is under oil immersion, 1,000 times magnified. So it is showing you one renal corpuscle, one corpuscle. Okay? What you see in the middle is the glomerulus. Okay? It's the glomerulus, glomerular capillaries. Okay? And then what you see, this outer lining that you see, it's a very thin layer of flat cells. That is the parietal layer of the glomerular capsule. And that white space that you see, the white space, that is the lumen. That is the space of the cup. So capsular space, you call it the capsular space. So figure B. So this is a slide showing you the renal cortex, where you see a lot of these. Okay, so we can move the slide and then you can see many of them. Okay, so this is the renal cortex. Now I'm going to show you C, the renal medulla, where you see these collecting ducts. The collecting ducts are lined by cuboidal epithelium, simple cuboidal epithelium. So I'm going to move the slide and show you the ducts. Here you go. So these are all the collecting ducts, and you can see the nuclei. I think it's much better on that TV monitors. And maybe if I go to 40, it might be better. Let me show you this one. Okay, these are the ducts. So this is the renal medulla showing you a cross section of the collecting ducts. So you can see this. As you move further, you might be able to see even more. So these are all ducts. These are all the collecting ducts. Okay, all lined by cuboidal epithelium. So for the microscope histology slides, you should know that B and C. Okay, B shows you renal cortex, the glomerulus, and the glomerular capsule in the renal cortex. C shows you the collecting ducts in the renal medulla. Okay, so these are the two slides. These are the two histology slides that you should know for the urinary system. And of course, like always, I will also have a picture of it. Now, the next thing that you're going to do is urine system physiology. Okay, if you can open up to page 183. Page 183. I have the same picture as well. How many of you are familiar with this? Okay, so this is a very quick way of analyzing urine for the presence of glucose or blood or protein. So this morning I told you proteins and blood do not get filtered, so normal urine should not have any protein or blood. Glucose is filtered at the glomerulus. It does get filtered because it's a small molecule, but because it's a useful substance in a healthy person, all the glucose is taken back. So urine should not have any glucose in it. As long as you're normal, urine should not show any glucose in it. Okay? So the test that we normally do, you can do it as a lab test as well, but very quickly you can test urine for quite a number of things. One is specific gravity. What is specific gravity? It is the weight of urine compared to water, or the density of urine compared to water. So pure water has nothing mixed in it. So water has a specific gravity of 1.000. Anything that has something dissolved in water will have a higher specific gravity. So specific gravity of urine is anywhere between 1.003 to 1.035. This is like a pH where you have no units. This is the same. You're comparing the weight of a solution to the weight of water. Blood will be even more heavier because blood has proteins and blood cells. It will be even more heavier than this. The specific gravity of blood will be 1.090. So it's going to be heavier. But urine, because it is water plus all the dissolved substances, it is slightly heavier than water. So our specific gravity can vary between 1.003 to 1.035 depending on what we have eaten. pH of urine can also be checked. So the more protein in your diet, the more acids in your urine. Okay? Urine will be acidic if you are on a very high meat diet. If you are a vegetarian and you are subsisting on vegetables and fruits, then your urine is alkaline. Okay? So the pH can be as acidic as 4.6 and alkaline as 8. This is the range. Your urine cannot become any more acidic than this. It cannot become any more alkaline than that. That's the range, 4.6 to 8. Color is pale yellow due to the presence of bilirubin. The characteristic smell of urine is because of urea, that is thrown out in urine. When urea comes in contact with atmospheric air, it gets converted to ammonia, and that's the smell. Urea getting converted to ammonia when it comes in contact with air. That means once you void urine, urea gets converted to ammonia. About 95% of urine is water. The rest is all dissolved substances. What is it that is dissolved? You'll have all salt, sodium, chloride, maybe a little bit of calcium depending on your diet. And definitely all the waste products. Uric acid, creatinine, urea. If you consume a lot of salt, then there will be salt in urine. Definitely no glucose and blood cells. Okay? Sometimes there could be trace amounts of amino acids. Especially bodybuilders, if you're consuming a lot of protein shakes and drinks, then some of the excess amino acids will be thrown out in urine because your body cannot absorb everything and use everything. Anything in excess will get thrown out. Okay, so when we do the lecture, I will tell you about this, why it is so. Okay, and then of course the volume of urine will change based on the liquid intake. So when you do some of these tests, you're actually checking to see whether a person, the urine is normal, or does it have any blood cells, does it have any glucose, and things like that. So these are called urine test reagents. Okay. So each one represents something: blood, protein, pH, ketones, okay, glucose. So each one represents something. So when you open the bottle and take out a strip, compare the normal color first. All of this means negative. Okay, each one, you have to go from top to bottom. The label on the bottle will tell you what each one is. Okay, and then we have fake urine samples. This is not real urine, these are fake urine samples. There are 3 of them. They're all labeled ABC. So dip one strip into the urine, take it out, and then shake it out a little bit so that the excess drips off, and then compare the color change to the chart. And then write down your results on page Write down the results on page 184. 184. You only have 3 samples, A, B and C. You can do this as a group. So I have separated it out into 7 test tube stands. So take one each and do it as a group. You don't have to do each one, doesn't have to do it. So take it, open the cap, put it in, try it out. So some of you already know how to do it. So this will also be there in your lab exam. So make sure you understand how to do it in 187. Number 1 to 9. Okay, those are just labeling and 187, answer question number 1 all the way to question number 9, and then from number 13 to 19, 13 to 19, and 21 to 24. So once more, number 1 to number 9, number 13 to number 19, 13 to 19, and then 21 to 24. That's it. You want more?
You want more?
Okay, next Monday is your lab exam. Okay? Next Monday.